We have our two sub-Is from the University of Washington who rotated giving the talk. The first one is Patrick. Patrick went to Rice University in Houston. He got a master of he got an undergraduate degree in bioengineering and then a master's, great engineering school. He's in his medical school here at the University of Washington. He's worked at some well-known labs at the University of Washington, the Grid Lab with Dr. Ojimin and Dr. Ko, and will do work next year, take a year off doing that kind of research. He had worked with Dr. Zhang and I in making nanoparticles way back when in 2020, right after COVID, actually. Anyway, without further ado, Patrick, what would tell us what you're talking on and anything interesting, any interesting perspective on your rotation? Did everybody treat you well?
Yes, and yeah, thank you for that kind introduction. Absolutely, I've had a wonderful time during this rotation. I feel like it really, um, just meeting every single, all the residents, all the mentors, all the faculty. It was really, really heartwarming and gratifying to know that such great people work here. And I've just had a great time.
It's all about mentorship. That's the fabric of what we do is mentorship. So I appreciate you saying that. All my hats off to all the people that teach in this university. So, Patrick, tell us what you're going to do. Tell us what your talk is about.
Absolutely. So I will just be giving a brief discussion on some of the research that I've been doing in the Grid Lab in the past, um, mainly focused on kind of the underlying neural activities and signatures associated with reward processing. Particularly, the task that I was looking into was something called EFERT, um, and that's kind of what I'll be talking about here today. So. Just to give a little bit of background for those who might not be as familiar with reward prediction or reward processing, just broad strokes, reward prediction or reward processing is a fairly instrumental aspect of motivated behavior, of motivated learning, and it's found a lot of application in terms of how we start to think about um, those who make decisions or have the inability to make motivated decisions. You can think of some clinical correlates such as depression, schizophrenia, some psychological diseases, but also there's other applications to motivated behavior and learning, most recently for large language models and AI and generative training, which have all found some use of understanding the underlying motivated behavior and reward processing learning. So that's kind of just broad strokes of what this is talking about. In terms of what we research and in this project is mainly correlated to neural features. So that would be features that we can see directly from instruments that we place or measure around the brain. The most common instrument, most useful instrument that we have, or I shouldn't say most useful, one of the main instruments that we have in particular is the EEG, which gives really great spatial and temporal resolution when we're trying to measure some small changes or deviations related to some sort of event. So these would be event-related potentials, also known as ERPs. And these become very useful because when you're trying to analyze a certain response or feedback in regards to some stimulus or reward, it's very easy to do on EEG because you can directly measure those electrical signals. Historically, there are a couple of main signatures that we look for in terms of control or reward-based learning. One of those is called feedback-related negativity. It's a particular signature in EEG recordings that occurs around 200 to 250 milliseconds after some sort of feedback or reward onset. And it typically shows up in the theta band range. So these are the particular signatures that are correlated with feedback related negativity. The other one that some people may be more familiar with is the P300. This occurs a little bit later. Um, it's more correlated with any sort of response to a stimulus, whether it's expected or unexpected. And these are typically a little bit slower in their wavelength, so that's more of the delta range. And also another tool that we have is functional MRIs, which can show direct um, impacts or direct activity in the brain uh, that is irrespective of EEG recordings. And this becomes very useful for very, very precise very, very precise spatial recognition of where exactly certain aspects of the brain are lighting up or becoming more active when you have some sort of learning or reward processing going on. I won't talk too much more about this in particular, but just know that it's a fairly powerful tool that a lot of researchers can also take advantage of when they're doing this type of research. Historically, I should also say that a lot of the research that's gone into reward processing have been mainly focused on the midbrain, including the tegmental area, the substantia nigra, the STN. But more recently, started expanding research and started discovering that the processes related to reward processing, which were previously thought of mainly dopamine or dopaminergic pathways, have these broad-ranging or supplementary aspects in other parts of the brain. So, for example, there have been some implications of, and I just put this on the right-hand side, of areas in the limbic system, for example, the insula, the amygdala, the hippocampus, which all seem to either also be related to reward processing or support the classical reward processing pathways that are seen mainly in the midbrain. And so when we're learning or trying to test um, reward processing, there are many ways to do this. There are a number of different tasks. And one of the, uh, or one that we've seen that is pretty well validated is called the Efort task. It's a task that is a little bit complex to explain, but I tried to break it up into its main components so that's a little bit easier to see. Essentially, a participant is asked prior to performing any task whether they want to choose an easy task or a hard task. And so they have to make that decision with really the only information being that they have a certain win probability. Now, this probability is random, so the patient will see either a win probability of 12%, 50%, or 88%. And then at that point, they get to decide whether they want to do an easy task or hard task. The two tasks themselves are related to pressing a button. The easy task is you have to press a button between 21 to 30 times with their dominant hand and their dominant finger or a dominant finger, while a hard task is they have to press a button for a much longer time using their non-dominant hand and a non-dominant finger. So it is a much more strenuous task when they have to do so. And then after they complete the task, they either win or they lose. And you can see that in terms of a reward probability or reward processing perspective, you can have a lot of different conditions. So if a patient was to have an unlikely chance of winning and then they ended up winning, you may expect that in terms of a reward prediction error, they might be more excited that they won because it was an activity that they weren't expected to win and they won. On the contrary, if they were expecting to win and then they didn't win or they lost, you might expect that that causes some sort of tension where a patient might be a little disappointed or frustrated that they didn't get the correct response that they felt like they might have deserved. So in doing this research, we collected so far 5 patients or subjects who were performing these tasks, and we were able to put stereotactic EEGs, mainly targeting various regions in the brain, but mainly looking at the limbic system to see if there were any neurosignatures that we could parse out that might help us better understand reward processing. Um, so in terms of just preliminary data and what we're able to see is that especially in the beta range, you can see that for what would consider a negative feedback mechanism, so this would be a case where a patient was expecting to win but ended up losing, there is a negative deflection in the beta range that occurs right between like 0 to 500 milliseconds, which is where you would expect to see, as I mentioned before, the feedback related negativity or the P300 neural signatures. So it is possible that this is an additional neurosignature related to reward processing. And I did look into uh, uh, other bands, other power bands, and it seems like beta was the primary one, if not the only one, that was implicated in the limbic system for these patients. Um, and so the next question then, oh, actually, before I go on to that, is where in the brain, when I mentioned the limbic system, were these results most apparent? And this is a heat map that we were able to create with just collating on a grand scale all the patient data and all the EEG data. And this is a little hard, but on the x-axis is time, on the y-axis is your channels or the areas of the brain. And then the heat map itself is how enriched a certain frequency is. So in this case, we're looking at beta. Blue would be lower enrichment, red would be higher enrichment. So this, as you're looking at it, we'll notice that in the temporal pole and in the hippocampus, you can see low deviations of beta, which is also what we had seen in the EEG recordings. So this also correlates kind of in the same time range to that, especially in the hippocampus and the temporal pole, not as much so in the insula or the amygdala. These are particular locations in the limbic system that we noticed these deflections in beta. And so then that brings up the question, obviously, of what does this mean? And I won't spend too much time on this, but just know that more recently there has been more research into beta power as potentially a neurosignature of motivated decision making. In particular, just recently, last year, there was a group that made a study looking into this specifically, and they had come to some observation that beta power, especially in the basal ganglia, had been related to effort and predictive effort in motivated decision making. And then there was also another one that the same group had put out that was also mentioning that not only was there differences in different areas of the brain and how they had different signatures in terms of um, different signatures in terms of how these brain activities were making, but also functionally connected. So, for example, an insular activity could then be coherent or impact somewhere in the prefrontal cortex. So all to say, in terms of discussion, the brain is very highly connected. It's so many things that we still don't understand. But in terms of reward processing and learning, it seems to be implicated in multiple domains that all seem to talk to each other, which is something we could expect. Future directions of this project that I'm interested in going into is kind of going along a coherence and seeing whether these limbic regions can talk to other regions of the brain, and then also using behavioral correlates and regression to further develop this research. So special thanks to the lab and everyone who's been working with this. I really appreciate all the work, and I really appreciate all the mentorship and help.
That's great. Super, Patrick, and really an active lab and about to implant their first brain computer interface device. So lots of exciting things going on in that lab. Questions, Patrick? We'll have time for questions now, and then we'll have Tom speak. So any questions for Jeff Heron?
Hey, Patrick, great talk, and it was wonderful working with you on a lot of this project. One of the things I'm curious about your take on is kind of like the role of stimulation as a kind of a scientific way to probe some of these effort decision-making circuits as well. Are there any if you were to pick some of the like the first target that you know of the targets you've been looking at for like stimulation to influence the outcomes of, is there anything kind of in your preliminary analysis kind of indicating that this might be a, one particular place or not might be an interesting place to stimulate to see how it affects the results that you have?
I would say one candidate I might think of is like the insula that immediately pops to mind. I just seen a recent paper where I think they did something similar where they had done an insular stimulation, um, which I think came out with some interesting result, but I can't quite remember what it was. But just from my analysis too, just looking at it, the main things that I was able to see is temporal pole, hippocampus, and then there was some insula that I wasn't able to show directly in the results here, but that would be maybe a primary target.
Cool. Thanks. Again, wonderful working with you.
Thank you.
Any other question? Great talk. Excellent. All right, was there another question? I'm sorry, I'm not seeing anybody in. All right, well, we're gonna go on to Tom Hanks, also University of Washington School of Medicine, gonna graduate in 26. He went undergraduate to Arizona State in Tempe. He got a Bachelor of Science in Exercise and Wellness. He's won a couple of awards, including the Kralik Foundation Scholarship. I didn't know Dr. Kralik started one, but that is great. You must be one of the first ones to get that. What's interesting about Tom, other than his name, is that he's an Alaska student. Isn't that correct, Tom? So Alaska student, and he's been working in Dr. Ferrara's lab. Anytime you go past the Ferrara lab, Tom is there working away. He's been very industrious. I can almost count on it. Nights, weekends. So fabulous work, and he's been working there since 2023. In fact, for 2 years. All right, Tom, tell us what you've been doing.
Yeah, well, thank you for the very kind introduction, Dr. Ellenbogen. Looking forward to sharing my work that I did in the Ferrara lab on chordomas for the past year. So we'll get started here. So no disclosures today. For some background, chordoma is an aggressive orphan tumor that originates from remnants of the embryonic notochord. Its incidence is 0.08 per 100,000, so it truly is a 1 in a million tumor. And the current standard of care includes maximally safe surgical resection followed by proton beam radiotherapy. But as the mechanisms of chordoma are poorly understood, there are currently no FDA-approved medical therapies for patients with more aggressive recurrent subtypes. So the primary objectives of today's talk are twofold. The first is to identify potential driver mutations within chordoma. And then the second is to assess the functional impact of these mutated genes in vitro with chordoma cell line models. So for our general experimental design, we performed whole exome sequencing on 49 chordoma tissue and paired blood samples at a read depth of 200x. Currently, this is well below any of the known chordoma series that have been published that utilize next generation sequencing. And then we ran these outputs through our bioinformatics pipeline that includes multiple variant callers that can help detect differences between the chordoma tissue DNA and then reference genomes that are involved in these pipelines. And to note, most of the published whole exome pipelines that are out there today include only 1 or 2 variant callers, whereas ours have 4 to 5, depending on the tissue type that's input into it. And then we visualize these results within RStudio and the various packages for that to help find the most common mutations and dysregulated pathways in chordoma. And then our current stage of experiment that's still ongoing includes an in vitro functional analysis of these genes. So next part of this, I'll dive into the bioinformatics analysis, some of those findings, and then we'll go on to the next portion there. So to assess if there were any mutational trends which led to more aggressive behavior in chordoma, we split up our cohort by non-recurrent and recurrent samples. And here we have a visual representation of the mutational summary within both of those cohorts, which shows an even distribution of variant subtypes and the types of mutations that were present. But notably within this, we did notice that the median number of variants per sample was quite a bit higher in the recurrent cohort. There were 24 at a median there compared to 9 within the non-recurrent cohorts. So it certainly indicates that there may be some higher genomic instability within these more aggressive subtypes. And then we took a look at the 20 most common mutations within our chordoma cohort. And we found that it was a fairly even distribution between the non-recurrent and recurrent subtypes. And the orange on the left is non-recurrent and on the right is the recurrent. And again, a fairly even distribution there. But the most notable finding for us was the presence of a very well-known tumor suppressor gene, PTEN, in 13 of our 49 samples. So that's over 25% of the cohort. And because this is such a well-known tumor suppressor gene that is present across many samples, we decided to do a deep dive into the potential implications of this gene in chordoma biology. So for some additional background, mutations in PTEN lead to unregulated cell proliferation. It's a hallmark tumor suppressor gene that's located on chromosome 10, and it negatively regulates the PI3K AKT signaling pathway, which is involved in multiple downstream pathways that are related to cell growth, proliferation, and survival. And then mutation of this gene has certainly been linked to unregulated cell proliferation and tumorigenesis in multiple other cancer types. Most notably for central nervous system cancers, it has been found in glioblastoma. And it has been reported within chordoma, but the true understanding of what this does has not been discovered yet. And so to validate this common variant, we performed pathway enrichment and gene ontology analyses, which help identify the mutated genes in our cohort that are significantly related to known biologic pathways. And interestingly here, the most significantly enriched pathway in our cohort was the PI3K AKT signaling pathway. And then if you look down the list there too, many of the others within the top 10 downstream or top 10 significant pathways are also downstream pathways of PI3K AKT. So that includes MAPK, focal adhesion, FOXO, and RAS signaling. And so this certainly confirms that it definitely has a significance in chordoma biology. So after this bioinformatics analysis, our primary question was then, how does PTEN impact the behavior of chordoma in vitro? And so this is where we decided to transfect mutant plasmids of PTEN into chordoma cell lines and go along from there to test its impact. So what we did here was we obtained 3 publicly available chordoma cell lines, all of which had been previously sequenced by the Chordoma Foundation and were confirmed to be wild type for PTEN. And then we designed plasmids which contained 2 different mutant PTEN isoforms that were found in our cohort and transfected them into the 3 chordoma cell lines. And again, the goal of this was to determine, does this impact chordoma behavior at all, proliferation? And if this is proven, could these mutants potentially be a target for drug therapy down the line? So as this is an ongoing stage of our study, this is more of a discussion about our early findings and can highlight some of the challenges that we faced in doing this. So in the upper picture there, you'll see bright field microscopy of our chordoma cells after several weeks of growth. And it highlights that there is a very prolonged time for these cells to reach confluency. And then below that is one of the GFP tags that we have attached to our transfected plasmids. Again, this demonstrates that there's likely some successful transfection, but probably only in a lower number of cells. And so in our troubleshooting process, we've been in contact with the Chordoma Foundation, and they've let us know that we are not the first to run into this obstacle. And I think it really speaks to not only the difficulty in treating this disease, but in also studying it in cell models. And so they suggested some alternative approaches with lentiviral transduction and potentially more stable given the latency of these cells. But there's pretty limited published data on that within the chordoma literature. So I think before we change directions, we're going to let this continue to run its course and then decide what else we can do from there. So in summary, we feel very confident that PTEN plays a role in chordoma biology. And as we perfect our transfection protocol and gain a better understanding of the function of PTEN and chordoma, We may gain some additional insights into targets for drug therapy and some treatment alternatives in the more aggressive subtypes. And then just want to give a few shout outs for people who had a really big impact on my research here. Of course, Dr. Ferreira, Dr. Parada for taking a flyer on me and letting me sink my teeth into a really big data set from the start and being patient with me as I figure out how this whole process works. And then Mark Diacenzo is the mastermind behind our pipeline, and he was very patient with me in the beginning as I was learning how to run a command line and figure out a lot of this bioinformatics process. And so very appreciative for all of them. And then my clinical research mentors, Dr. Rusvik, Dr. Emerson, getting to work with them in the skull-based group. And, they've provided me a lot of good guidance and feedback over the last year. More recently, working with Dr. Mayberg on the patch clamp project and getting to see, a different side of research, but very exciting to be involved in that. And then, of course, my resident mentors, Zach, Dom, and Malia, spent a lot of time with them as they were all on research in the past year, and very exciting to see, what that looks like a couple years down the road, and to get to learn from all of them. I think it made the year a lot more fun. So, with that, I'm happy to take any questions.
Any questions? Ali Ravenpay has his hands up, Tom.
Hey Tom, I don't want to make this sound easy, but going back to what you were talking about in terms of transfiction, was there any thought given to maybe establishing primary cell lines from non-recurrent tumors? Because it sounds like you have some challenges with using even the established cell lines. I'm wondering whether you could kind of just bypass that idea and maybe get a little bit even more relevant answers after you do the transfiction.
That's a really good question. And, we spent a lot of time wrestling with how we wanted to approach our in vitro analysis. And the short answer to that is getting these cells to grow even with established transfection protocols is extremely difficult. And we have obviously a massive tumor bank repository and absolutely samples that we know contain this PTEN mutation, but we spoke with Chordoma Foundation about that, and even getting that off the ground is not just a months-long process, it's years potentially to establish these cell lines. And so I think there's absolutely, a merit to doing that in the future. But I think with the constraints that we had on time, we decided to go with these previously established ones, just as there were some known protocols. And as we can see, even with that, it's very challenging to do.